Hello and welcome to BioRules video lessons. Today I'm going to be taking a look on the structure of proteins. So you carry on what we were studying last class, which are the amino acids and gluing the amino acids together to be making polypeptides or proteins. So just a reminder, this is condensation. I have amino acid 1 on the left, amino acid 2 on the right with radical 1, radical 2. And for me to do the condensation, I take out water. Here it is, the water that was taken out. And I bond the two amino acids with a covalent bond known as peptide bond. So basically, if you want to make a protein, if you want to make any kind of protein, you are going to get one amino acid and you're going to put together with another amino acid. This here, the condensation process will make a peptide bond. And when you have that, you don't have an amino acid anymore, you have a G-peptide. Peptide is a name for protein. G-peptide, two of them together. And if I glue another one, so amino acid 3 now, I just made another peptide bond, and now I have a 3-peptide. And if I connect many of them, n amino acids over there, I'm going to be ending up having a polypeptide. So a polypeptide is the same idea as a polysaccharide, many sugars glued together. Polypeptide, many amino acids glued together, making a string of amino acids that can be a protein in the future. So polypeptides, they are one of the names that we give for proteins. They are not totally synonym, but you will get the idea, okay? So a sequence of amino acids will make a polypeptide. So depending on which amino acid I put one beside the other, I make a polypeptide. And this is what we're going to be calling the primary structure of a protein. A protein has a certain sequence of amino acids. Depending on which amino acid you have a protein, and if you change that sequence, you have another protein. So if you have methionine, valine, histidine, glycine, it's different if you have methionine, glycine, valine, histidine. So if you change the sequence, you change the protein. So this is the most basic idea of what is a protein. A protein is the sequence of amino acids that it has. It will determine all the other structures as well. And what makes this here to happen, the sequence, is the peptide bond. What glues one amino acid of the other is the peptide bond. Of course, the information to tell your body which amino acid to put one after the other, this is a much more complex idea that we're going to be studying later. Amazing stuff. But for the moment, what makes a protein is what are the amino acids in sequence one after the other. Okay, so a few examples here for you of different primary structures. So you have the glycine, proline, tryptophan, glycine. You have a certain sequence, one primary structure. Here you have another one, glycine, isoleucine, valine, cysteine. You have another primary structure. So primary structure, which amino acid goes side by side in the string of amino acids of a polypeptide or of a protein. Here is the idea of a primary structure, which one goes after the other. Okay, if there's a primary structure, there is a secondary structure. What's the secondary structure of a protein? Things start to become a little bit more tricky. The secondary structure is the folding or the coiling of the polypeptide basic structure. What's the polypeptide basic structure? It's the NCC, 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 and so on. So if we ignore the radicals, if you forget about the radicals over there, the NCC, NCC, NCC itself, the very basic structure that is the same independently on which amino acid you're putting over there, this sequence over there may fold itself and may coil itself, making a secondary structure. Okay, so the radicals are not going to take part on the secondary structure here. What will make this folding or coiling is hydrogen bonds between the hydrogen of the amino group and an oxygen in another amino acid, an oxygen of the carboxyla in another amino acid way ahead. In this example here, I'm taking the third amino acid and the first amino acid, and we have the hydrogen in the amino group of the first amino acid and the oxygen and carboxyl of the third amino acid, and voila, they are connected by hydrogen bonds. Of course, they get close together to be able to do this, but this is just for you to visualize the idea. Let me show you a secondary structure. Here we have a secondary structure. On the right, you have all the chemicals there together, and you are probably able to see in dark red here, I'm going to put here, we have many hydrogen bonds. These are hydrogen bonds between the oxygen of the carboxyl and the nitrogen of the amino way ahead. Here we have, look, hydrogen of the amino and an oxygen in the fifth or sixth carbon amino acid ahead. So you have here and here and here and so on. They are connected by hydrogen bonds. And when they do that, in spite of the radicals, no matter what are the radicals that are there, this protein is coiling itself in a alpha helix. It's coiling over there. And you're going to be having hanging out here the radicals. Radical there, radical here, radical there. The radicals are hanging out. They don't take part on this stuff here. They just made a coiling over there. Here, two examples for you. As I mentioned before, the alpha helix, that makes this coiling of the protein, but it also may make a beta pleated sheet, also known as beta plate or beta sheet, but beta pleated sheet is the best name, where you have a little plate of protein that have been folded, making this little plate over there. Also secondary structure. So here you have alpha helix on the left, beta pleated sheet on the right. Those are examples of secondary structure. As you can see, the radical is hanging there. You see there are radicals over here, they're hanging, but they're not taking part on the structure, on the secondary structure. See here, radicals over there, they are hanging there, but they're not taking part on the structure of the protein. And here we have, now remember the primary structure, the sequence which one goes after the other, and here you have an alpha helix, and here you have a beta plate making secondary structure. Okay? There's a secondary structure, there is a tertiary structure. Yes, tertiary structure is very important. Tertiary structure actually is going to be absolutely important because it's going to make the protein to work. Okay? The tertiary structure is a three-dimensional folding of the protein. Proteins are strings of amino acids that may be coiled or plated or whatever as a secondary structure, but in the end, they end up folding, making a three-dimensional structure. Proteins have a shape, and this shape gives it the function. So it happens because of the radicals. Now the radicals are going to be interacting between each other, and they are going to be making the protein to fold. This image here is going to be illustrating the way that the radicals can make the protein to fold over each other. Okay, there are 4 different types of bonds that can work in here. One is ionic bonds. If you have an acidic amino acid in one side and an alkali amino acid on the other side, no, positive and negative, they may get attracted to each other depending on the pH of the solution, depending on many things, but they may get attracted to each other, no, attaching one side of the string to the other. Okay, we may also have a G-sulfide bridge. As I showed you before, cysteine has sulfur, and 2 amino acids with cysteine, a cysteine on one side and a cysteine on the other side, they may glue here by a G-sulfide bridge, making a very strong covalent bond between 2 sides. Another possibility is to have hydrogen bonds. So if you have here an amino acid have a hydrogen with an oxygen, and here a double oxygen, you may have here a hydrogen bond, and they might get close together, no, bonded. And you, last but not least, we may have a hydrophobic interaction, 2 hydrophobic apolar amino acids. If this protein is in water, these amino acids are going to be running away from water, and they will end up getting together, and by getting together they may up, helping to stabilize the protein structure over here in a 3-dimensional shape. So I hope you remember there are some amino acids that are apolar, so all of those can make hydrophobic interactions. Some of them are polar, so they're not going to be making hydrophobic interactions. Some of them are acidic, some are alkali, and if you remember cysteine over here, the one that has sulfur, it can make disulfide bridge. So depending on the amino acid, you have different radicals, and depending on the radical, you're going to have a connection of the protein, connecting themselves in different ways. And when they connect themselves in different ways, they make connections here that stabilize the protein with a very specific three-dimensional shape. This is how the radicals help to give the protein a three-dimensional shape that will give in the end the protein its function. Here we have primary structure, secondary structure, and then this can all coil around making a full three-dimensional shape of a protein, tertiary structure. Oh, is it over? No, not yet. It might not be over because some proteins may have a quaternary structure. Ooh, what is it? We already have a three-dimensional shape. Is this the fourth dimension? No. Actually, some proteins, they make a polypeptide that have a three-dimensional shape, and they work, they make their function beautiful, it can be okay. Some proteins have only three dimensions, no primary, secondary, and tertiary dimension, because they are made of only one polypeptide. But some proteins that are actually made by connecting different numbers of polypeptides together. In this example here, we have two subunits, so two different polypeptides that get connected together and they make a different shape. So in this case, we have one protein made by two polypeptides. So the idea of quaternary structure is when you have more than one subunit attached and working together, making a function. Who is responsible for that? It's going to be the radicals as well, okay? So the same bonds that make the tertiary structure, they can also make the quaternary structure. They may bond the subunits together, okay? Not all proteins have that, so let me show you here an example. We have on the left myoglobin. This is a protein that's capable of storing oxygen in muscle. And on the right, we have hemoglobin, a protein that holds and carries oxygen in blood, in the red blood cells. The myoglobin is made of one subunit, so it has only tertiary structure. But hemoglobin is made of 4 subunits, so it has 4 polypeptides, so it has a quaternary structure over there. Okay, so tertiary is the maximum on the hemoglobin, and because made of one subunit, and in the myoglobin, sorry, on the left, and the hemoglobin has quaternary structure and it has 4 subunits. Another example of proteins that have quaternary structure, antibodies. Antibodies are formed usually by 4 polypeptides. They make together this way shape, so they have 4 polypeptides glued together, working together, making a three-dimensional shape that you have a function, so they have quaternary structure, okay? Antibodies, if you're not aware, they're made for defense, and they also have 4 subunits, so quaternary structure. So here is the idea. When you put the subunits together, hey, 4 subunits over there, you're going to end up having quaternary structure, okay? So primary structure, secondary structure, tertiary structure, and then quaternary structure over here. I think this is quite straightforward, and I hope you're not going to be having trouble with that. Okay, one more thing. Proteins can have a slight variation being fibrous or globular. Okay, so on the left you have an example for fibrous protein, collagen. It has a quaternary structure because it's made of 3 polypeptides that now get interlaced together. And on the right we have another quaternary structure, a hemoglobin that I showed you before, of 4 subunits. What's the difference between fibrous proteins and globular protein? Globular proteins, usually they are globular. Oh yeah, they are kind of roundish. Now they have a 3-dimensional clear shape. And fibrous protein, usually they are straight and long. And another difference that fibrous protein, usually they are insoluble, and globular protein, usually they are soluble. But they are proteins, they work in different cases over there. And in some cases, it's difficult to tell if it's a globular or fibrous protein. So it's more like extreme examples. Collagen is an extreme example of fibrous protein, and hemoglobin is an extreme example for globular protein. Okay? I hope you had fun and you got the idea of the class. And do the quiz right now, and I'll see you again in the next bioresidual lessons. Bye bye.